Hello, and thank you for tuning in to this webinar. My name is Dr. Jeffrey Wood, and I'm an assistant research professor here in the School of Natural Resources at the University of Missouri. I wanted to take this time to introduce myself before I disappear behind the slides that I'll present to you, showing some of the research that's been conducted at the MOflux site, which is located at the Basket Wildlife Research and Education Center. So without further ado, I'm going to switch over to my deck of slides and we can get underway. The topic for this webinar concerns research on the drought impacts on Missouri's Oak-Hickory forests. Before I begin, I'd like to take this time to acknowledge the generous funding support provided by the U.S. Department of Energy, Office of Science, through Oak Ridge National Laboratory's Terrestrial Ecosystem Science Focus Area. I'd also like to acknowledge collaborators Leon Hungu and Paul Hansen of Oak Ridge National Lab, as well as Steve Pallardy from the University of Missouri. So first, a little bit of background for the webinar. I'm sure it's not a surprise to anyone, seasonal drought is quite common in Missouri. Here's an image showing the US drought monitor map for the state of Missouri, just at the end of that severe drought in 2012. And while we don't always see drought this severe over the entire state, usually in each year, somewhere in Missouri, there will be some period of drought. However, we often hear about the impacts in terms of how the drought affects croplands. For instance, if you were to Google drought in Missouri, most of the results would be on how drought has reduced crop yields and farm income, for instance. However, there is also a lot of forest in Missouri, and drought is also affecting those areas. And so we're interested in learning about how drought is affecting the functioning and growth of these forests. We can actually, just with our own eyes, see that drought has a huge impact on the forest. And so you can see that here in these two photographs on the right. The top photograph was taken at Basket on September 3rd of 2010, after a very wet year. And you can see how lush and green the forest looks. In contrast, the bottom picture was taken on September 2nd in 2012 after that very severe drought. And it looks like it's fall colors, maybe mid to late October, early November. But this is actually the top of the canopy being scorched during the drought. And so just, again, this is an illustration with our own eyes. We can see that the drought can have a profound effect on these forests. And what we do is study in much more detail how the whole forest, down to the leaves, stems, and roots, are being affected by these droughts. So a little background on the MOflux project. So MOflux is short for the Missouri Ozark Ameriflux site. And this project was started in 2004 by scientists from the University of Missouri and the U.S. Department of Energy's Oak Ridge National Laboratory. This is a long-term project, and the overall goal is to understand how drought and extreme weather events impact eastern U.S. deciduous forests. We use sophisticated sensors as well as standard forestry methods to study this forest from roots to shoots and leaf to landscape. We have sensors that are mounted on the MOflux tower, which you can see here on the right in two of these photos. This is a tower that's about 1 3/4 times the canopy height. And these sensors measure the flows of carbon dioxide, water, and energy between the forest and the atmosphere. So you can think of this as we're measuring the breathing of the forest. We also have vegetation plots for tracking forest inventory, tree mortality, as well as litter production. And we also make a lot of physiological measurements like leaf gas exchange. And you can see that being done here using a photosynthesis system. And so with this collection of measurements, we can really understand at very fine process scale, as well as whole ecosystem scale, how drought impacts this forest. And that is the subject of this presentation. And generally, we're going to be focusing on water stress and impacts on the breathing of the forest. If you'd like to learn more about the MOflux site, you can check out our virtual site tour video that you can find elsewhere on this virtual field day. So first, let's talk about water stress and how we measure that and the observed variability. So we use a variable called pre-dawn leaf water potential to track water stress in different species in the forest. And I'm showing you here some of those data in these 3 panels on the right-hand side. In each panel, the vertical axis presents the pre-dawn leaf water potential. And on the horizontal axis, we're showing time as represented by the day of year. In each panel, the way you interpret these numbers is such that when the values are close to 0 here at the top of the panel, that's indicative of very low water stress, or no water stress, I should say. And as we decrease in pre-dawn leaf water potential, that's indicative of increasing stress. And down here at -4 megapascals, that is very, very stressed indeed. Each panel is showing 6 different lines, and each of those different lines corresponds to a different tree species. And you can see those different species in the legend here. The main takeaway from these plots should be that we see between years, so if you compare from one panel to the next, we see a very different pattern of pre-dawn leaf water potential and thus water stress. Everywhere from very wet conditions where all of the species are tracking about the community mean because there's no stress, through situations of intermediate drought where the relative drought adaptations of these species become evident, and you can see that by the separation of the different species. So white oak is quite a drought tolerant species in comparison to white ash, the red line here. However, we also see that exceptional drought condition where, again, all of the species are tracking together about the mean because it's become so dry that it has overwhelmed those different drought adaptations, and everybody's suffering and unhappy. So we see quite a lot of variation from year to year, as you see across panels, as well as you can see how dynamic and variable the water stress situation is within a year. So now let's take a look at how we can observe, or what we've found in terms of water stress and its relationship with precipitation variability. So to do this, we first need to calculate some metric for integrated water stress. And we use something called the PLW-PI. And this is a measure of the accumulated water stress over the growing season. And so this is accounting for both the duration of stress and intensity of the stress. And the interpretation of this PLW-PI is shown here in this graph in the bottom left, where you can see we again have pre-dawn leaf water potential on our vertical axis, day of year on our horizontal axis. And in this case, we're showing the community mean pre-dawn leaf water potential. And that's this black line that you see here on the graph. And this should look familiar from that bottom panel in the prior slide. And the PLW-PI is simply the area between that seasonal curve and our 0 line here. And so this is giving us, again, an accumulated metric of water stress for that growing season. And we can then take that PLW-PI and compare it to statistics that describe precipitation variability. So here I'm showing you that in terms of PLW-PI on our vertical axis related to mean daily precipitation rate here on our horizontal axis. So our horizontal axis spans from dry on the left to wet on the right, and our PLW-PI or stress axis spans from low stress at the top to high stress at the bottom. And as you can see, we plot all of our years or growing seasons up as these blue points, and we can fit a line through those points and find a very tight relationship that tells us that simply mean daily precipitation rate explains a lot of the variation from year to year in water stress. So now we've seen how we measure and track water stress, and that it's very dynamic, both across years and within years, and that much of this water stress variability is controlled by patterns of precipitation. So now let's look at how water stress exerts controls on the breathing of the forest. And so to look at that, we'll look at the CO2 exchange rates, and we'll use a variable called the net ecosystem CO2 exchange, or NEE for short. And NEE is the balance between the ecosystem respiration and ecosystem photosynthesis. So that's the difference between the CO2 release and the CO2 uptake. So I'm showing you on the right-hand side of this slide, again, a representation of our water stress situation, again showing here in the upper panel, pre-dawn leaf water potential on our vertical axis, day of year on our horizontal axis. And so again, remember the less negative or the closer to zero reflects low stress, more negative, getting down to the bottom of the panel, that means high stress. So again, we have our 3 wet drought and exceptional drought years. And in this representation, these are areas. And so at any given time slice, the vertical distance that you see on that area represents the range of observed pre-dawn water potentials across all the species. So the difference between the maximum and minimum observed values on that day. On the bottom here, you can see our net ecosystem CO2 exchange, or NEE, plotted on our vertical axis and day of year on our horizontal axis. And so in this panel of showing our NEE dynamics, when you see positive numbers up here near the top of the panel, that means that the forest was a carbon source. So it was breathing out or exhaling CO2. And when you see negative values, like particularly between these vertical dashed lines, when you see negative values, that means the forest was a carbon sink. So it was breathing in more CO2. And so what I really want to highlight is that water stress varies significantly within and across years. And if you compare the top and the bottom panels here, you can see that it exerts strong controls on NEE dynamics. And so one of the big effects you can see here is if we look at our NEE plots, this blue year, a wet year, shows us kind of what a reference sort of unstressed situation looks like, where early in the growing season, there's this precipitous drop to peak CO2 uptake, and then this kind of gentle shoulder on the senescing side of the annual cycle. And the big effect that our droughts have here in Missouri is compressing the duration of that net carbon uptake period. So you'll notice that the width of the time period here where it's spending below the zero or in the negative numbers, you see that width decreases as we go into the gray drought year, and particularly during that exceptional drought year. Well, this could be a bit depressing because you see we, in a drought year, can go carbon neutral, where we basically switch off the sink and respiration and photosynthesis is in balance. And in exceptional drought, we can go to a situation where the forest is actually a persistent source of CO2. And this was for about 50 days in July and August, which is very impressive and severe. While that could be a bit depressing, we do see that if there are soaking rains, the forest can recover quite quickly, provided that those rains occur before too far into senescence. And you see that here in that exceptional drought year, we recover very rapidly to about where we would expect to be during a wet year. So it's showing remarkable resilience when we get that rehydration after rainfall. And you can see that reflected in our water stress measurements or our pre-dawn data up here in the top panel. That rehydration event in the wet year, or in that pardon me, exceptional drought year was quite profound, and you see that here, showing that return to a much more hydrated status. While we do see this very strong effect on, and influence on the dynamic of NEE, it's important to note that even during that exceptional drought year, on an annual basis, the forest was a net carbon sink. And so here in this table at the bottom of the slide, I'm showing you the annual NEE, which is basically just summing up over the whole year for those 3 years. And you can see we were about 200 grams of carbon per square meter even in that exceptional drought year. And so while we don't totally fail, it is considerably less than in a wetter year situation. Now, one of the issues is if you were to have this situation identical every year, where you started to have major drought severely limiting, and you kind of have this accumulated buildup over time, that's when we could get into a much more precarious situation. But thus far, because of this strong variation in drought from year to year, we haven't seen any temporal trends in or time trends in NEE over time. However, that's not to say that we haven't observed important effects of the drought in a much shorter time frame that's more significant, but this is at the individual level, where we've observed significant tree mortality following drought. And so one of the things we found after that major drought in 2012, which occurred here in this graph of species stem mortality over time, a huge pulse in 2013, where stem mortality increased quite dramatically, particularly for white oak in magenta here, and black oak, which was about 25% stem mortality in that year, which is rather significant. Given what I had presented earlier, where we showed that the oaks tended to be more drought adapted, one might ask, well, why is this happening to our oaks? And we found evidence that there was a drought pathogen interaction whereby weakened oaks became very susceptible to a fungus called Biscognoxia, formerly known as hypoxylin, which infected the trees, and it was affecting the oaks, but not the other species. And so Biscognoxia gets in and rots the sapwood, and you can see in this photograph here a stem that's been covered in Biscognoxia. It causes the bark to slough off, and this rotting of the sapwood essentially comes in and finishes off the tree after that severe stress. And in our inventory, we found that of the trees that died in 2013 for white oak, both white oak and black oak, about 75% of those trees showed visual evidence of Biscognoxia infection. A couple of other important points: as I noted, these were weakened trees that tended to die. So the trees in the inventory that tended to be showing lower growth rates before the drought tended to be the ones that died, versus ones that were showing higher growth before drought. Another thing is that we found no evidence that this mortality pulse had a detectable or noticeable effect on the CO2 balance of the forest. And so we haven't seen catastrophic drought effects in terms of massive die-off or alterations to ecosystem functioning that were persistent over many years. So that concludes the new material for this talk. Before I wrap things up, I'd like to highlight a few key points. So one, MOflux is a long-term project dedicated to better understanding the impacts of drought and extreme weather on deciduous forests more broadly, but also specifically in Missouri. We observe substantial variation in water stress, and this exerts, in turn, strong control on the carbon balance of these forests. However, we do note that the forests can be quite resilient and show remarkable recovery if there are soaking rains before senescence. The decrease even though we see strong decrease in carbon during the drought, even in the driest year in record, the forest still was a sink for CO2. Finally, we do see elevated tree mortality that has lagged sort of a year after the drought and preferentially affected our oaks because of a drought pathogen interaction. And although we do see these tree mortality pulses, we have not found evidence that it's substantially altered our net ecosystem CO2 exchange in terms of a time trend. If you have more questions, I'd be happy to entertain those, and you can reach me either by email or in my office, and my contact information is provided here. I'd particularly recommend email at this time, given the status of the pandemic. And for those interested readers, you can check out some of the published papers that I'm showing you here in this reference list, where some of the results that you've seen today have been published previously. This last publication is actually a bonus feature. I did not show any of those results today for the sake of time, but as a teaser, that was what happened in this forest when the lights turned out in the middle of the day during the total solar eclipse of 2017. So thank you again for your attention and tuning in to this webinar. And I hope you've enjoyed this first virtual field day for Basket. And hopefully we can see you sometime in the future in person at Basket and MOflux for the real thing, shall we say.